Vidyapith and the entire team of Banasthali for inviting me here to talk about this cell culture. So, uh, and I keep on coming to the institute because I find this institute is doing a great job towards the women empowerment, and they have a very dedicated team of faculty, staff, and all. So whenever I feel like, whenever they call me, I feel very honored to be here. So thank you for the ability. Please continue like this. So and I think this will actually help in building the nation. So okay, good morning everybody. So now today I will talk about one important aspect of animal biotechnology, that is advanced cell culture techniques. And as you all know, animal biotechnology base or fundamentals of animal biotechnology is the cell culture practice. So we cannot imagine the drug discovery program. We cannot imagine development of novel molecules. We cannot imagine anything coming commercial product coming out for the consumption of human being without the use of animal-based or cell-based models. So we have two types of models. One is cell-based model and there is animal-based model. So prior to animal-based model, we have to use the cell-based model, which is more cheaper and more authentic models to be used. So today I have divided my talk into two parts. First part I will talk about the culture of animal cells. So first I will start with 2-dimension, then I will move to 3-dimensional culture, and then I will end my talk with some aspects of stem cell biology, induced pluripotent stem cells. I will complete that. And I will show you some of our experiments that we are doing to give you a glimpse. And I heard from Vidyapith that some of you are our PhD students as well. So I will try to show you how you can design experiments so that you can use them for your research also. Very simple experiments I will show, and which you can also design in your own labs to improve your quality of your research. Okay. So so this is the introduction. So what is a cell culture? So you all know that our body is made up of about 37 trillion cells. All the organs that you see in our body are made up of cells. Organ, tissue, whatever you talk about, they are all made up of cells. So many cells comes together to make organs. Many organs comes together to make an organ system. Okay. So when I say cell culture, what I am meaning that I am taking the cells out of the body and trying to propagate them outside the body. And that condition, what is required for the survival of the cells outside the body, is known as the cell culture condition. Okay. So that condition, knowing what condition will allow you to grow your cells in the in vitro condition, is known as the culture of cells, or in other words, cell culture or tissue culture. Okay. So now if I see, this is how you can see here. This is, say, for example, a embryo, say chicken embryo. Now what is there? It is made up of millions of trillions of cells. And now if I so when I separate these cells out individually and I try to culture them in the in vitro condition using a culture medium and the cells. So as you can see here, these are the cells, and the upper layer that you see here is the culture medium. What is a medium? You all know it. Medium is the fluid part which actually contains all the nutrients required for the growth of the cells. So now the cells will be here, and the cells will, if they are here, they will try to proliferate, means cell division takes place, and then the plates gets full, and that's how cells remains in this condition like this. And you can continue culturing these cells in the in vitro condition, days after days, months after months, years after years. Now, why do we culture these cells? I will talk about that. Why we need to culture these cells? Why we need to propagate these cells? So, basic concept is that cells has to be isolated and they can be cultured in the in vitro condition, and that is what we mean for cell culture techniques. So now, if I see the origin of a cell, what cell comes from in our body? So if you see, our life starts from a very simple organ. We all are originating from a sperms and eggs fertilized together to make a zygote. And zygote is an unicellular structure. We all came from one single cellular structure called zygote. And zygote is basically a totipotent stem cell. It's also a stem cell, and you all know what the totipotent stem cell is. Totipotent stem cell is that toti means total, potentiality means to form. So a cell which can make a complete organism is known as totipotent stem cell. So we all came from a totipotent stem cell. So one zygote divides 2 cell, 4 cell, 8 cell, 16, 32 million trillion cells, and all the organs of our body has come from one that particular single cell that we got, that is the totipotent stem cell. So we all started our journey of our life from a zygote. So what is that? That means in our body we have stem cells. So totipotent stem cell zygote, then zygote divides 2 cell, 4 cell, 8 cell and so on. Soon it loses its totipotent shape. Means, and say for example a zygote has divided 3 times or 4 times. So it has now become 16 cell or 32 cell stage. And if I split the cells apart, those cells will not make any complete organism, but they can make all sorts of tissues and organs of our body. That means we will not no more call those cells as totipotent stem cells, we will call them as pluripotent stem cells, because they cannot make a complete organism, but they can make all cells and tissues of our organism. So this is the story of a cell. So if I see a stem cell, this is a stem cell, say for example pluripotent stem cell. Now what will happen? The pluripotent stem cell is destined to form a particular cell or tissue of our body. Okay, it is going to form that particular. So before it forms, and that cell or tissue is known as the differentiated cell. Say for example, a stem cell become liver cell, hepatocytes. If it becomes liver cells, what will happen? The cell will differentiate to the liver cell. So if it's that is the differentiated product is the differentiated liver cell. Okay. So now in between journey from stem cell to a differentiated liver cell, it covers some in between stages known as progenitor cell. So this is basically the progenitor cell. So progenitor is the intermediate stage between a stem cell and a differentiated cell. So what is the property of this cell? The property of the cell is neither liver cell, neither a complete stem cell, but it is gradually moving towards the differentiated cell. So you have to understand that cellular differentiation, stem cells differentiation does not happen overnight. Today is stem cell, tomorrow it's differentiated cell. It happens a gradual process. So this gradual process becomes stem cell, progenitor cell and the differentiated cells. So this is how the cell progresses from one form to another form. Now you have to remember one important thing, that stem cell, when they remains there, so one of the important property of stem cell is self-renewal. What is self-renewal? They proliferate within itself. Why do they proliferate? They make a population of cells. Why do they need a population of cells? Because once the cell differentiates to differentiated cell, so it will not divide anymore. Differentiated cells never proliferate. So naturally, if your liver gets damaged for permanently or is a permanent damage, you cannot replace it. But liver damage happens, but we repair it out. Why do we repair it out? Because there are a small pool of cells, stem cells, who are retained there. How they retain there? So whenever stem cells differentiates to liver cells, some cells remains there as a small pool of cells. Whenever there is a damage in the future, those cells will again proliferate, they will become progenitor cell, they will differentiate to, oh, this is smart structure. That's what I was thinking while changing itself. So these cells will change back to this and then they will differentiate. So that is why the property of stem cell is the self-renewal is a very critical property. That means it always keeps a pool of it. And as you grow old, the pool of those stem cells are gone. That's why our regeneration process is also less. So when you are young, if a bone fracture happens, it repairs in a few weeks. If a kid, even the doctor goes and you don't put a plaster, you just say, okay, it's fine, it will cure automatically. At this your age, you need a plaster. But if somebody 80 years old, bone fracture happens, doctor said nothing can be done. If it heals, it doesn't heal. Why? Because by the time the pool of stem cell in that 80 years old person is already gone. There is no stem cell pools, and if it is not there, so there is no more for cells to become progenitor cell, no more cells differentiate to cells. So as you grow old, the differentiation pattern changes, and that's why stem cell undergoes one process is called. So that is why the stem cells, one property is self-renewal, and the property is differentiation. So stem cell has two functions, self-renewal and self-differentiation. Now, whenever you culture the cells are cultured in two different conditions. Two types of conditions are there. One type of cells are known as adherent cells, and that is known as the suspension cells. What is the difference between these? Adherent cells means they will adhere to some matrix surface. Tissue culture plate. If I take stem cells and try to grow it in the tissue culture plates, you will find the cells will go and attach to the matrix surface or the culture plate surface, and they will have a distinct shape. So the cells which need a matrix surface for the attachment are known as the adherent cells, and the cells which does not need the attachment, they remain suspended and they will not have any shape. They will be spherical or circular in shape. Who are those cells who will be suspended, who will be adherent cells? The cells which in our body, because you know, cells when they are in the in vitro condition, they maintain the same condition as it is in the body. 37 degree temperature, 20% oxygen, and humidity is required, pH is required, everything what is there in the body, that is to be there in the in vitro condition. So which cells will remain suspended in the in vitro culture conditions? Some blood cells, like blood cells, RBCWs platelets. If you are using for your experiments blood cells, they will never be adherent cells. They will always be suspended cells, because in the body also the cells, blood cells remain suspended in the blood. So plasma and all they floats around. So this you can see cell lines which is adherent here, so suspended here. So these are blood cells. But other cells like liver, heart, kidney, lungs, they are not suspended cells. They are all adherent cells because they are attached to each other. They are tightly packed to each other. That's why those cells will be known as the adherent cells. So they will remain adherent to that, and they will have a distinct shapes. So basically, classically, we divide the cells into two forms: adherent cells and the suspended cells. Okay. So based on the shapes of cells, the adherent cells are divided into four different categories. Fibroblastic cells, very elongated structures like cells. There could be epithelial cells, which will be little bit not spherical exactly, say hexagonal type pattern. They will have a hexagonal like structures, epithelial cells. There could be another class of cells known as endothelial cells, which makes our blood vessels of our body. Endothelial cells, which shape would be somewhere less elongated as these cells and not as rounded as these cells, but in between structures you can see here. So they are the endothelial cells. And there is the neuronal cells. And neuronal cells are the easiest cells to identify because they will have neuronal projections. You can see axon and dendrites could be seen. Whenever you culture the neuronal cells, they will be visible like this only whenever you culture these cells. So broadly, we culture cells that are adherent cells because most of the cells are adherent cells. So they will have this type of four patterns: fibroblastic cells, epithelial cells, endothelial cells, and neuronal cells. So these are the four different cell types that are generally used. Now, why do the cells adhere to the matrix? Why the cells needs an attachment to the matrix surface? Why the cells are not remain free in the culture conditions? So this is the basic diagram which shows how the cells behaves. If you see here, this is a cell attaching to the, and if you have this line that is shown over here indicating the matrix surface, means tissue culture plate surface. Okay, so now this is a cell, this is a cellular nucleus, and in the cell, this purple colored thing that you see, they are basically the membrane protein known as integrins. Integrins are surface membrane proteins of any animal cells. Okay, so what happens? These proteins actually helps the cells to anchor to the matrix surface. If you remove this integrin protein from the matrix surface, the cells will remain free. Protein condition will not adhere to the matrix surface. So it is adherent to the matrix surface. Now, what will happen? As the cells keeps on attaching to the matrix surface better and better, so what will happen? These integrin proteins starts to dimerize. So usually they are monomer, but they then dimerizes to come together. So you can see here the integrin proteins are dimerizing. And when the cells are attached best, you can see the cell is more spread. So that means that this cell is attached better way to the matrix surface as compared to this one. Its shape is changing. So this cell has changed its shape from here to here, and it is more spread like structure. Because as I told you, the adherent cells will have a distinct shape because they adhere to the matrix surface very strongly. So if they remain attached to the matrix surface, so they now once they are spread, what will happen? Dimerization of integrin protein has taken place. So that is an indication the cell is broadly attached to the matrix surface. Now, after that, what will happen? These cells, which are adherent cells, they will not grow well if they are suspended. Why not? This is shown over here. Because once the cell attached to the matrix surface, it activates some signaling molecule. I am not sure if you are aware of these signaling molecules or not. One important molecule I am talking about is FAK, focal adhesion kinase. Focal means a particular point, adhesion means adhering to the matrix surface, kinase means a protein which phosphorylates other proteins. So, as soon as the integrin dimerizes and attaches very nicely to the matrix surface, this pathway, these proteins gets activated. And you have to understand, these proteins are linked to the proliferation and differentiation of the cells. So if I inhibit the interaction of these cells with the matrix surface, what will happen? This pathway will be not there. And this pathway not there, this cell will remain like this. It will not divide, it will not differentiate. So you have to provide a proper matrix surface for the cells to attach, and if they attaches to the matrix surface,